Hey everybody, Dr. Rafich here, Vet Triage. Wednesday, October 23rd, 2:03 p.m. Pacific time. Boy, I do not look good in this light. I'm at my friend's clinic today helping out, but gotta do a live stream. So we're going through this issue of JAVMA. It is January 15, 2016 edition, and we're talking about the effects of processing delay, temperature, and transport tube type on results of quantitative bacterial culture of canine urine. And this comes out of Texas A&M and a specialty hospital in Florida. The objective is to determine the impact of processing delay, temperature, and transport tube type on the results of quantitative bacterial culture of canine urine. And what they did was they took urine aliquots. They were spiked with bacteria. They used E. coli, and then they measured how much E. coli grows from those samples. So the results were the median bacterial concentration for urine samples stored in a urine transport tube for 24 hours at 4 degrees Celsius was lower than that of samples stored in a silicone-coated clot tube under the same conditions. Conversely, a decrease in median bacterial concentration was identified for samples stored for 24 hours in a silicone-coated clot tube at 25 degrees Celsius, compared with the concentration of samples stored with the urinary transport tube under the same conditions. The median bacterial concentration in samples stored in a silicone-coated clot tube at 25 degrees Celsius for 24 hours was less than the cutoff typically used to define clinically important bacteriuria, which is bacteria in the urine, by use of urine samples obtained by cystocentesis. And typically you want to see bacterial concentrations of over 1,000 CFUs per milliliter, which is colony-forming units per milliliter. So in conclusion, the canine urine samples submitted for immediate quantitative bacterial culture should be transported in plain sterile tubes, such as silicone-coated tubes. When it's prolonged, meaning 24 hours storage at room temperature, then urine samples should be transported in urine transport tubes. So if you're going to transport, if the sample is going to wait 24 hours at room temperature, then you want to transport it in urine transport tubes. If you are sampling them in an ideal situation, meaning right away for bacterial culture, then you want to use the silicone-coated clot tubes. Okay, let's go into the introduction. Approximately 14% of dogs will have at least one UTI, that's urinary tract infection, during their lifetime. The gold standard is bacterial culture of urine. The International Society of Companion Animal Infectious Diseases recommends that the aerobic bacterial culture and susceptibility testing be performed on urine collected by cystocentesis from animals with simple or complicated UTIs. Cystocentesis is a transabdominal approach. It's minimally invasive. It's the same as a blood draw, except that the needle is going in through the belly, from the belly into the urinary bladder, and the urine is removed. And that's as sterile as you possibly can collect. Sample handling affects culture results, and optimal quantitative bacteria concentration results are consistently obtained when urine samples are processed immediately after collection. So, for example, you grab the sample of urine and you place it in, they're inoculated onto agar plates, and then that grows the bacteria. The problem is, in a real life situation, most hospitals, including this one, they have to submit their urine to the lab. They have to transport it. So you're not getting it right from the patient into the agar plate. It has to be transported. Samples are often collected and held for courier pickup or shipped overnight. Urine samples stored in plain sterile tubes at room temperature for 24 hours prior to plate inoculation are diagnostically unreliable, with false positives seen in 50% of samples. To mitigate the deleterious effects of storage, duration, and temperature on quantitative bacterial culture results, tubes contain chemical preservatives to try and combat this. And these preservatives provide an attractive alternative to plain sterile tubes. Commercially available tubes contain a buffered boric acid mixture, which contains sodium formate, sodium borate, and boric acid, and they have been validated for urine storage in human medicine. According to the manufacturer, urine samples may be stored in room temperature in such tubes for up to 48 hours without compromising test results, and refrigeration will not affect the results either. The objective of this study was to compare bacterial concentrations in sterile urine samples that were spiked with E. coli, transferred to traditional silicone-coated tubes or urinary or urine transport tubes, and stored for 0, 8, or 24 hours at 25 degrees Celsius, which is about 77 degree Fahrenheit, so that's room temperature, and in 4 degrees Celsius, which is refrigeration, or 39.2 degrees Fahrenheit. So materials and methods. Urine was collected via catheterization or manual expression. A total urine volume of 200 milliliters was collected, pooled and stored overnight at 4 degrees Celsius. The urine was then filtered to become sterile twice. The sterile urine is then inoculated with bacteria. Bacterial counts were performed by making a 10-fold dilution series of urine, inoculating 100 microliters of each dilution into the blood agar plate, which contains trypticase, incubating the plate overnight, so about 18 hours, at 37 degrees Celsius, and manually counting the colonies. Bacterial suspensions were 1 milliliter of urine from each of the 6 aliquots. They were transferred into 5 plain silicone-coated tubes and 5 urine transport tubes. And the urine transport tubes contain the preservative, which is a combination of sodium formate, sodium borate, and boric acid. Tubes were allocated to be processed for quantitative bacterial culture immediately, which is 0 hours, or stored for 8 hours, or stored for 24 hours prior to inoculation. Tubes allocated to be stored for 8 hours or 24 hours were separated into 2 groups, ones that were stored at 25 degrees Celsius, which is room temperature, or one at 4 degrees Celsius, which is refrigeration. Bacterial concentrations were then estimated. Trypticase soy agar supplemented with 5% sheep's blood, which is the blood agar plate, was used. The plates were intubated overnight, 18 hours at 37 degrees Celsius in 5% carbon dioxide. Colonies were manually counted the next day. Results. No significant differences in bacterial concentration were identified for urine samples stored in either tubes at 4 or 25 degrees Celsius for 0 or 8 hours. However, significant differences were evident for samples stored at 24 hours. Bacterial concentrations in urine samples stored for 24 hours at 4 degrees Celsius, so refrigeration, in urine tubes were significantly lower than those stored in the same temperature, same duration for silicone coated. Additionally, after 24 hours of storage at 25 degrees Celsius, there was a significant decrease in bacterial concentration in samples stored in silicone tubes compared with the urine tubes. A significant decrease in bacterial concentration associated with storage duration and temperature when silicone-coated tubes were used was seen. Post-test results indicate a significant difference in bacterial concentration between urine samples stored for 0 hours and those stored for 24 hours at 25 degrees, as well as stored at 8 hours at 25 degrees Celsius, and those stored for 24 hours at 25 degrees Celsius. Evaluation of urine samples stored in urine tubes at the 2 temperatures revealed a significant decrease in bacteria over time. Post-test results indicate significant differences between urine samples stored at 0 hours and those stored at 24 hours at 4 degrees or 25 degrees Celsius. Finally, bacterial concentrations in urine samples stored for 24 hours at 25 degrees Celsius were lower than those stored for 8 hours at 4 degrees Celsius. So, discussion, results. The results of the present study indicate that regardless of storage temperature, the concentration of E. coli, and this is strain ATCC25922 for those that care, but anyway, the results indicate that regardless of storage temperature, the amount of E. coli in urine samples decreased significantly over a 24-hour period when samples are stored in the silicon or urinary tubes. Most importantly, the bacteria concentration in urine samples stored in silicone tubes for 24 hours at 25 degrees Celsius was lower than the numbers considered clinically important to consider a UTI, which is 1,000 colony forming units per milliliter. And this potentially results in a failure to identify an active UTI. However, for urinary tubes, the median bacterial concentration was greater than that of the clinical limit in urine samples stored for 24 hours at either 4 degrees or 25 degrees, and no significant difference was identified in bacterial concentration between urine samples at 4 degrees and 25 degrees Celsius. The results indicate that urine samples from dogs stored in silicone or urine tubes may be left at room temperature or kept refrigerated for up to 8 hours without changes in bacterial concentration. That's up to 8 hours. 8. But urine samples stored in silicone tubes and refrigerated for 24 hours may be reliably used for quantitative bacterial culture. And that's more likely to be the case when you're sending a sample out to the lab. Quantification of the amount of bacteria in a urine sample allows veterinary practitioners to distinguish between a true infection versus specimen contamination. It also allows objective differentiation of UTI relapse from reinfection. The clinical importance of a positive culture result is influenced by the sample collection method. For example, a bacteria concentration of 3,000 colony forming units per milliliter would indicate infection if the associated urine sample was collected sterilely via cystocentesis. However, for a naturally voided sample, this result would most likely represent contamination, and diagnosis of UTI requires over 10,000 colony forming units per milliliter. Because urine is a viable culture medium, boric acid is a suitable preservative for urine samples. The borate ion has bacteriostatic properties, maintaining a stable environment for bacterial growth while limiting contaminant overgrowth. So bacteriostatic means there's a difference between bacteriostatic and bactericidal. Bactericidal means that it kills the bacteria. Bacteriostatic means it doesn't kill it, but it freezes the bacteria from replicating further. One study revealed that boric acid was a satisfactory urine preservative, given that it maintained the existing microbiologic flora without yielding high numbers of false positive results. On the contrary, in a UK study, addition of boric acid resulted in an increase in the possibility of false negative results. So we need more studies to assess preservative type. Limitations of the study, and so this is where my thoughts were. They only tested E. coli, and that's because E. coli is the most common bacteria implicated in urinary tract infections. However, they only account for a third of all the isolates. Gram-positive cocci, like Staphylococcus and Streptococcus species, are the second most common. And then less common organisms include Proteus species, Pseudomonas species, Mycoplasma species. In most circumstances, there's a single bacteria that's causing the UTI. Another limitation of this study is that urine samples were collected from dogs without a UTI. I'm also going to mention that the samples were voided from the dogs or catheterized. It wasn't done with cystocentesis, which is considered the gold standard way of collecting urine. And then finally, they only tested dogs in this study. They didn't test other species. Their tubes were filled with only 1 milliliter of urine, and the manufacturer recommends 4 milliliters. That also is another limitation of this study. And I don't know why they only collect a sample from like a handful of dogs. So I'm wondering if this study was done in conjunction with another study of why those dogs were euthanized or whatever, because it's very easy to collect urine. You can get a much larger sample size than this. So they had a very small sample size of urine to begin with, and they had to partition that out for all these tests. It's a bit weird. The results reported here suggest that canine urine samples should be promptly placed in a silicone-coated tube, then stored and shipped at 4 degrees Celsius if you're going to process it immediately. Or if you can't process it immediately. So if you can't process it immediately, then the urine samples should be placed. Let me repeat that. If you're going to evaluate the samples immediately, you want them transported in a silicone-coated tube. If, however, that's not possible. And you can store and ship them at 4 degrees Celsius if you can't do immediate processing. And that's the majority of clinics. When canine urine samples are likely to be exposed to an ambient temperature during storage or transport to the laboratory, then you want to actually use a urine tube. So a urine tube, if the sample is going to stay for in room temperature during storage or transport, a silicone tube if you're going to sample the urine right away. And if you can't send it right away, you can keep it in the silicone tube, stored and shipped at 4 degrees Celsius. So that's the study. Hopefully that's helpful. There's a lot more to be done, of course, in this realm, but that's a pretty good start to how to direct doctors in processing the urine samples for UTIs, which is very common. So thanks everybody for watching. I'll see you guys again tomorrow, October 24th. Thanks for those who watch in the playback and who donate and subscribe. We appreciate it. Take care. Bye.